ते फिशरीच्या मुलांची तेवढी लिस्ट आणून द्या ना. हा निकिता कर सुरू. अगं हे इथे जे दिसतंय ना स्लाइड शो तो कर, म्हणजे तुझी ही जी प्रेझेंटेशन आहे ना हे प्रेझेंटेशन मोठं दिसेल आपल्याला. इथे वर, इथे वर वर वर. बरं होत नसेल तर असू दे, जशा कंडिशन मध्ये तसं सुरू कर.
चालेल ना.
ओके.
My name is BSc 3. My seminar topic is dairy product. In this topic, I will tell you about dairy products like dahi, ghee, khoa, which we use in our daily life. Ma'am, shall I begin?
हो का, कर सुरू.
A variety of milk product in India. We consume a variety of milk product in our daily life, in which cheese, butter, condensed milk, milk powder, etc. we eat, like milk. Dairy farming is highly attractive industry. Many peoples are getting employed in dairy farming. The first dairy product is dahi. We consume it in daily life in India. Dahi is very famous in our kitchen. It is mainly used for salad and in diet also. Dahi is prepared by souring milk with lactic acid starter, which is usually curd of the previous day. In process of making curd, we usually use the curd of previous day because it is concentrated with organisms like streptococcus and lactobacilli. The organism present in the curd are mainly streptococcus thermophilus and lactobacilli casei. The curd is normally consumed at the place of production because curd is get expired or get uh, contaminated during the 2 to 3 days. So generally curd is prepared in kitchen and it is eaten at the place of production. Next is khoa. Khoa is eaten in as prepared or it is consumed as it is because it is prepared by rapid evaporation of water from the milk. That means when we get 1 liter of milk and when we evaporate it, only 2 ml of the khoa is remained. Buffalo milk is used for the khoa preparation because in buffalo milk fat content is more. Kava is directly consumed or used as an ingredient of sweet. All of you know that we use kava in the preparation of many sweets like peda and other sweet recipes. It can be kept for 3 to 4 days. Ghee or clarified butter is obtained from the butter by eliminating water. In the process of making ghee, we get butter on the pan and we eliminate water by heating it for 5 to 10 minutes. It is very important dairy product can be stored over long period of time. The ghee of cow is yellow while the buffalo is whitish in color. We can store ghee for the long period of time and the ghee of buffalo is generally white but the cow ghee is uh, yellowish color. Generally in kitchen, ghee is used to cooking different sweets and recipes like frying, jalebi, etc. And it is also taken directly on the food like rice. Butter is a mixture of milk, fat, buttermilk, and water. In butter, taste is salt and coloring materials are used. It is good source of vitamin A and D. We all know that butter is we directly use in food recipes like street food and homemade food like paratha. The last product is cheese. Cheese is product made from the curd obtained from the whole or skimmed milk with or without added cream by coagulating the casein, and further the separated curd is treated by the ripening of fruits. We generally see that cheese and cheese slices are used on the sandwiches and other burgers, etc. The soft cheese is also called as paneer, and we use it for the paneer tikka and other recipes of paneer. Thank you.
चल नाईस प्रेझेंटेशन. नेक्स्ट हर्षल. हर्षल कदम. तयार आहे का रे तुझं हर्षल? चल कर सुरू.
कालचा तो हॉरिबल होता. काय माहिती नाही. मी म्हणलं
पीपीटी प्रेझेंट कर तुझी. हा
गोळे बिळे
मॅम दिसते का?
हो दिसते. कर सुरू.
Good afternoon, everyone. Myself Harshal Kadam. My seminar topic is pearl culture. In pearl culture, there are sub-points are following. First is introduction. Second, production of pearl. Third is chemical composition of pearl. And fourth is stages in pearl formation. Introduction. The pearl is also known as moti. We all know moti. A round concretion of skeleton material, biochemical products of mollusk, lipids and fatty acids, etc. Only them highly shining globular in shape. Shining globular in shape. Very shining pearls. Pearl consists of a central nucleus surrounded by a series of pearl nacreous matter. Pearl is surrounded by a nacreous matter. चमक पदार्थ असतो ना, असं pearl surround केलेला असतो. Nacre mother of pearl is a shiny, hard, pearly, iridescent substance forming the inner layer of mollusca shell. This shell consists of 3 layers, namely an outer organic concave and 2 inner calcareous layer. Next point is production of pearl. There are 2 ways of production of pearls. First is natural way and second is artificial way. In natural way, natural pearls formed due to natural process or disturbance in environment where the oyster lives. The second one is artificial way. In artificial way, artificial inducing the mollusk to secrete nacreous matter around an inserted nucleus. It is produced by the human interference and is known as cultured pearl. Cheap imitation is made of plastic that have an artificial luster are known as artificial pearl. In artificial pearl, human interference is involved. Next point is chemical composition of pearl. In pearl, there is a 3.97% water is present, 3.87% organic matter is present, and 91.53% calcite and aragonite is present. Quality of pearl determined by its keratin content and color. The latter is influenced by the color of shell, donor mollusk, from which pallium had been taken and the part from where it is extracted and implanted. The next point is stages in the pearl formation. There are 6 stages in pearl formation. First one is preparation of host shell. Second is nucleus insertion. Third is convalescence. Fourth is pearl formation. Fifth, harvesting. And last one is processing of pearl. The first stage is preparation of host shell. In nature, pearl may be formed in different regions of the body, namely the region of adductor muscle, mental margin, and pallial zone. Pearl production is to region, 20 individuals good nucleus is inserted in good region. Next stage is nucleus insertion. In nucleus insertion, involves or any other object for obtaining the nuclear material and donor oyster or muscle obtaining grafted tissue. The size of the nucleus determines the size of the grafted tissue. After obtaining the nucleus grafted tissue, the oyster will be operated upon and the nucleus implanted in the gonadal region. The time and the number of nuclei to be implanted will be determined in advance. Next stage is convalescence. The nuclear insertions, after nuclear insertions, the muscles are allowed to be recovered from the disturbance caused. The oyster are kept in cages. Next stage is pearl formation. The graft tissue on the nucleus grown into sac. The epithelial cells of the pearl sac deposit nacreous layer around the nucleus, which ultimately leads to the formation of pearl. Next point is in pearl oyster, it takes 3 to 4 years of commercial value to develop. In freshwater mussels, pearls are harvested 2 to 3 years after implantation. Next stage is harvesting. Oyster or mussel with pearl are brought to the laboratory. The valves are opened and the fresh parts are extracted out. The pearls are obtained by pulverizing the extracted part. The next stage is processing of the pearl. Soon after their separation from the meat, pearls are washed with water to remove mucilaginous matter and the luster. The pearls are then graded according to their size. Thank you. मॅम झालं मॅम
हा बोल ना.
आपलं
बोल. संपलं पण का? प्रोसेसिंग ऑफ पर्ल नाही का? पर्ल फॉर्मेशन मध्ये कुठल्या मेथड्स यूज करतात सांग?
प्रोसेसिंग ऑफ
पर्ल फॉर्मेशन मध्ये कुठल्या मेथड्स यूज करतात? आहे का काही मेथड? स्टेजेस मध्ये जी न्यूक्लियर स्टेज सांगितली आहे तू, न्यूक्लियर आहे का न्यूक्लियस आहे?
न्यूक्लियस
इन्सर्शन. त्यामध्ये काय होतं? पर्लचं फॉर्मेशन कसं होतं न्यूक्लियस मध्ये?
नाही आता तुमचं माप मला कळलंय. त्या मापाचं पाहिजे आहे की नाही?
बरं ठीक आहे. नेक्स्ट
ते कसं होतं काय
नेक्स्ट कोण आहे?
बांगडीच्या ह्याच्यावरती
रोहित.
ठरतं ते माप.
रोहित माने प्रिपेअर आहे का?
ही बांगडी मला बसते, प्राजक्ताला बसत नाही ही बांगडी.
रोहित माने.
तिचा हात मोठा आहे.
स्वप्नील गोडसे.
मला 2.25 गाळा आहे. तिचा 2.5 आहे.
हा रोहित सांग, तयार आहे का? स्वप्नील बरं कर, पीपीटी शेअर कर आणि सुरू कर. होत नाहीये का स्क्रीन शेअर? रोहित
हो असे असतात.
तुझी स्क्रीन शेअर कर. आणि सगळ्यांनी आता सेमिनार झाले की ज्या फीडबॅकच्या लिंक तुम्हाला पाठवलेल्या आहेत ते फीडबॅकच्या लिंक आज भरणं कंपल्सरी आहे तुम्हाला. आणि भरून झाल्यावरती लगेच स्क्रीनशॉट पाठवा मला ग्रुपवर की मॅडम आमचा भरून झालाय म्हणून. आणि बाकीचे अजून जॉईन झाले नाही त्यांना सेमिनार द्यायचा नाही का काय?
मॅम प्रज्वल अडकलंय ते दरड कुठं कोसळली ना वाईच्या तिकडे अडकलंय ते.
बर.
त्याने काल दिलेला निरोप त्याला ते कनेक्टिव्हिटी प्रॉब्लेम.
ओके. स्वप्नील तुझी पीपीटी शेअर कर आणि सुरू कर तू. स्वप्नील गोडसे आहे का स्वप्नील?
सोन्याच्या बांगड्या पण घालून जाते का कंटाळा येतो.
काय झालं रे रोहित? रोहित, अरे तुझी स्क्रीन दिसत नाहीये.
मॅम नेटवर्क प्रॉब्लेम येतोय.
बरं मग थोड्या वेळाने कर तू. दुसरं कोणीतरी घेऊ तोपर्यंत, ठीक आहे?
हा चालेल.
स्क्रीन शेअरिंग ऑफ कर तुझं. दुसरा कोण आहे? स्वप्नील, शिवानी आवडे तुझं राहिलेलं आहे का?
हो मॅम.
बरं कर सुरू. मग तुझी स्क्रीन कर शेअर. रोहित तुझं शेअरिंग स्टॉप कर, त्याच्याशिवाय तिला करता येणार नाही.
केलंय स्टॉप मॅम.
पण इथे तू दिसतोयस ना समोर मला प्रेझेंटिंग करताना. हा शिवानी सुरू कर. शिवानी, आवाज येतोय का माझा? हॅलो हॅलो, अरे बोला, आवाज येतोय का? स्वप्नील, ऋषाली गाडगे,
हा येतोय मॅम.
तुझं राहिलेलं आहे का सेमिनार?
नाही झालाय माझा काल.
बरं. प्राजक्ता,
येस मॅम.
तुझा राहिलेला आहे का?
हा राहिलाय.
झालाय का?
हो झालाय मॅम.
बरं. स्वप्नील, स्वप्नील गोडसे,
हॅलो मिस, माझं राहिलेलं आहे.
बरं कर, तुझी स्क्रीन कर, प्रेझेंट कर आणि सुरू कर पीपीटी.
फक्त आहे असं वाचायचं. स्टार्ट शेअरिंग कर. स्टार्ट कर.
काय झालंय ग? जिथे तुझी पीपीटी ठेवलेली आहे ती पीपीटी ऑन कर. आता तुझं स्क्रीन शेअर झालेली आहे. प्राजक्ता,
हा येस मिस. मॅडम स्क्रीन दिसते का मिस?
आता नाही दिसत आहे, याच्या आधी दिसत होती. कारण तू तिथे जिथे तू तुझी पीपीटी ओपन केलेली आहे तीच दाखव.
हा एक मिनिट.
हा हा दाखव. कर सुरू, दिसते आता.
Good afternoon, everyone. My name is Prajakta Nandakumar Nagre. My topic name is seminar disposal and pollution of pharmaceuticals. Paper number PCC, Immunology. Introduction. As the world pollution contains increase the expand rapidly, they our daily in life food and drugs are important in modern generation in human lives. Many diseases are occurs they way that's why are required the production from many pharmaceuticals drugs and products. After use of pharmaceutical drugs and products may pharmaceutical waste are developed, and due to this we require disposal and pollutions control pharmaceuticals. Definition of pharmaceuticals waste, in which includes used and unused expired preparation pharmaceuticals products, home use, personal care products, and our of the uh, controlled medicines have emerged sense of developments of standard medical waste regulation of being a new major public and environmental health. Next one, types of pharmaceutical waste. First, chemical waste. It includes in pharmaceuticals waste like outdated, contaminated, undiscovered medicine. Second, biomedical waste. Waste from laboratory culture, stroke, uh, our specimen of microorganism, leaves or attends vaccine, human and animal cell culture used in research in industrial agent from research and industrial laboratory. Waste from the production of biological toxin and dishes and device used for transfer of culture. Sharp waste includes needles, scalpels, blades, and sickles. Third, other pharmaceuticals मध्ये soiled, semi-soiled and products, tablets, capsules, granules, power for injection, mixture, creams, lotion, gels and suspension. Liquids मध्ये solution, suspension and sprays etc. आणि मध्ये injections, spray and inhaler. Second point, causes of pharmaceuticals products. Healthcare industry, hospital and nursing home also contribute of pharmaceuticals pollution. Hospital in polyculture might be loss of problem as they have on-site pharmaceutical nursing and home other uh, pharmaceutical guilty of filtering medicine down the to our dairy. Drug manufacturing. Mm, Some factories are bigger than others. They are guilty of cultures of pharmaceutical pharmacy pollution. Third, agriculture and agro product. The drugs and hormones we are meant to make the livestock and poultry grow fast will be keeping from guilty sticks. As such as those hormones and antibiotic will lead to growth water. Human human domestic drugs used and disposal behavior. The pharmaceuticals and personal care products that end up to streams, groundwater, lakes, and rivers. That's next point is pharmaceutical waste. त्यामध्ये expired pharmaceuticals, second contaminated pharmaceuticals आणि third pendant pharmaceuticals. Infection waste, lab culture waste from isolation wall, tissue swabs and materials requirement of injection patients. Sharp waste, needles, infused sets, scalpels, always blades and broken glasses. Toxic waste contains cyclo drugs and used in cancer therapy. Chemical waste, lab recent, expired disposement, expired solvents. Third, effects of pharmaceutical pollution. Effect of fish and aquaculture life. Some toxic chemical substances are affected by fish. The fish species of can affected to these toxic chemicals. In strong chemicals effects of male fish and female fish ratio. Second, effect on drinking water. In drinking water sources like river which can from agriculture, domestic and industrial sources such as pharmaceutical contaminated this water. Third, long term affection of the environment. Some pharmaceutical company last than time in the environment and wastewater supplies. These chemicals pollution waste from stream, lakes and rivers. Fourth, effect on wildlife. Pharmaceutical fishing into the environmental by humans thus has affected the wildlife. Second point, pharmaceutical waste disposal method. Any product requires disposal solution entirely spreaded from packaging and example glass, plastic bottle, other contents by destruction. Risks associated with destruction of the product, potential for product of get divided during disposal sources and construction of growth water. Disposal produce should be involved agents. What is prevent recovered to dealing with such circulating material and company plan to disposal. Next point, pharmaceutical solid and liquids waste and disposal management. Soil waste material like injection tablets, capsule, hand gloves, ampoules, spraying, whistles, salines, plastic bottle, etc. Disposal of material with proper technique. Disposal of packing completed post health risk. Such material
आणखी
should be.
किती राहिलाय ग प्राजक्ता?
एक पॉइंट राहिलाय मिस.
मग तो ना म्हणजे तुझ्या भाषेत सांग, फक्त वाचून दाखवू नकोस. कन्क्लुजन काय आहे सांग त्याचं मला.
Yes, they refuse to disposal can be not solved which plastic bag in induction divided participate required means
जोरात बोला, आवाज येत नाहीये.
government should be play important to disposal of waste economically. This thus educated and emotion of self first important is important and the pages of other about it. Start disposing waste first from which you with you your home and outside home. They nitrogen they are yourself stick to city and then nitrogen world. Let's make them worlds better plus place to live.
अभिजित तांबे.
हा मॅडम.
हा कर, तुझी पीपीटी शेअर कर आणि स्टार्ट कर.
हा.
प्राजक्ता तुझं शेअरिंग स्टॉप कर ग.
तुमचे पण बघा शेवटचे
रोहित.
एंट्री दिल्या का फक्त शेवटचे दिले ना एंट्री.
रोहित
हा मॅम.
का तुला नेटवर्क आलंय का?
हा आले.
सगळं करून दिले ना.
हो सगळ्या जेवढ्या बसल्या.
दिल की बघ.
हो.
एक वर्ष.
तांबे अभिजित विश्वास. एक कुडवे म्हणून एक आहे. स्लाइड शो कर. हे बघ इथं वरती आरोय ना अभिजित. बाकी काय करू नको. वरती एक चौकोनात आरोय बघ. एकच पीपीटी सगळ्यांनी घेतली. एकच पीपीटी आहे. नॅपिकलच्या तुम्हाला पण एक कॉमनच आहे ना. कॉमनच आहे. हा.
जमदाडे मॅडम. हॅलो हॅलो.
हा बोला मॅडम.
हा मी रेकॉर्डिंग स्टॉप करते, तुम्ही तिकडनं स्टार्ट करा.
हा ठीक आहे. अभिजित स्टार्ट कर तू.